What's up, y'all? Welcome back to another episode of Purple Bear Biology, where we discuss biology, anatomy, and microbiology. If any of those topics sound awesome to you, then consider clicking the subscribe button so you never miss an episode. In this episode, we are going to explore how we can predict whether a population is stable or evolving, and how we can directly measure evolution in a population over time. The last thing we'll talk about are a couple different types of selection that can take place that can lead to a population's allele frequencies changing over time. Wait, what was that word again? Allele? What was an allele again? Alleles are versions of a particular gene, like the gene here that codes for flower color. For this particular gene, there's a red allele and a white allele. Genotypes are the sets of alleles that we get from our parents. Alleles can be dominant, where they mask the presence of a recessive allele, or codominant, where both alleles get expressed at the same time. But we'll talk about that here in just a little bit. First, we're going to discuss the following 5 conditions that can cause a population to evolve. Populations that are evolving have changing allele frequencies and are said to be out of Hardy-Weinberg equilibrium. So let's talk about these 5 conditions that can cause allele frequencies to change over time. Let's get started with mutation. We have already seen through different examples that mutations can lead to genetic changes in a population over time. But let's use this example of a parasitoid and a singing cricket. No doubt you have heard crickets at night singing away. Ah, the sweet sound of romance. Male crickets sing to attract mates, but females looking for love are not the only ones that hear this sweet serenade. There's a parasitoid fly that uses crickets as its host that can also use these calls to find the males. The fly lays eggs inside of the cricket, and the young eat the cricket from the inside out until bursting out like tiny little aliens. That's a pretty horrific way to go. There is, however, a mutation that causes male crickets to be mute and not sing. Now, under normal circumstances, this mutation would be incredibly detrimental because the singers get all of the ladies. But when the environment has a serial killer parasitoid, it's best to be the quiet one. So you see, when the environment favors a mutation, the mutation will become more common in a population as it gets passed along to the offspring. This results in allele frequency changes in that population as a whole. Now let's take a look at natural selection. Remember that organisms are born with specific adaptations, and when those adaptations give the individual a higher chance of survival and reproduction, then they become more common in the population as a whole. This too leads to allele frequency changes over time, which causes the population to shift out of Hardy-Weinberg equilibrium and begin evolving. So those two are what we've talked about before, but now let's discuss something new. Let's talk about genetic drift. Genetic drift occurs when random events cause population allele frequencies to change over time. This could be due to like a random reduction in a population, or like random pollination of flowers. Either way, allele frequencies are changing, so evolution is occurring. However, it is not due to the fitness of the allele. This is just chance luck that results in a skewed allele frequency in the next generation. Another common condition that causes evolution is something called gene flow. Gene flow is essentially migration. When organisms from one population migrate into another, this can change the proportion of the alleles present, sometimes even introducing entirely new alleles. Because the alleles are changing over time, the population is evolving. Again, this is not really about relative fitness of one allele over another, but rather changes in frequencies due to modifications to the original population. The last condition we will discuss is non-random mating. When mates choose their reproductive partners based on preferences, this can lead to one allele being more preferred than another. This type of choosiness can be seen in birds particularly, where coloration and feather quality can be predictors of whether or not a male will get a mate. Technically speaking, we have 2 types of preferences that can take place. Intrasexual selection, where males compete. This is kind of like where male deer fight over territory, and intersexual selection where females are choosy and males get to mate based on female preference. I should mention that in some species, it's the males that get to do the choosing. This type of selection can be pretty cool. I'll give you this example. The organisms that you see here are called stalk-eyed flies. The males actually have their eyes out at the end of these long stalks. Females prefer longer stalks, and so males line up and allow females to compare stalk length to determine which she would like to mate with. Selection like this can lead to dramatic differences in the morphology between males and females in a population. and these are referred to as sexual dimorphisms. Collectively, one or all of these conditions may be acting on a population and resulting in allele frequency changes. But how can we know? How can we determine if a population is out of Hardy-Weinberg equilibrium? We can calculate allele frequencies and determine what genotype frequencies should be in a population if it's in equilibrium. If the actual genotype frequencies do not match what we would predict to be the genotype frequencies in the population, then we know that it's out of equilibrium and is evolving. To calculate predicted genotype frequencies, we will use the Hardy-Weinberg equilibrium equation, which is a mathematical representation of these genotype frequencies. P and Q represent the two separate allele frequencies for a given gene, where conventionally we use P to represent the dominant allele and Q to represent the recessive allele. In codominant scenarios, it really doesn't matter as long as you keep your two alleles separate and give P to one of the alleles and Q to the other. If I start talking about P's and Q's and squareds all together without providing you an example, you're probably going to get lost pretty quick. So the easiest way to do this would be to take the Hardy-Weinberg equilibrium equation that we have here and solve an example. Keep in mind that P squared, 2PQ, and Q squared all represent your genotype frequencies that you would predict a population would have if it's in equilibrium. P squared would represent your homozygote dominant individuals, Q squared would represent your homozygote recessive individuals, and 2PQ would represent your heterozygote individuals. Now let's put this all into practice, using this population as an example. Here you can see we have a population of roses. These flowers come in 3 color varieties, red, pink, and white. In our example here, this coloration is going to be created by a gene that has 2 alleles, a red allele and a white allele. Together, these 2 alleles exhibit a codominance where both red and white get expressed as pink. So let's talk about how we can determine if this population is in equilibrium. We have to start by calculating the actual genotype frequencies. If there are 500 individuals in the population, then we can divide each of these phenotypes by the total number to get the frequencies in the population. Because this is a codominant scenario, our phenotypes and our genotypes will match. Dividing the prevalence of each of these phenotypes by 500 gives us the actual genotype frequencies of 64%, 32%, and 4%. Next, what we want to do is calculate the frequency of each individual allele, the red allele and the white allele. To do this, we must take the total number of homozygote individuals and multiply it by 2, because each individual has 2 copies of the allele, and homozygote individuals have the same copy. We then need to add the number of heterozygote individuals, because each one has a copy of each allele. We can do this for both of our alleles. We have 320 red flowers, which would be our homozygote dominant, multiplied by 2, because each of them have 2 copies of that allele, plus our heterozygote individuals, each having 1 copy of the red allele, giving us 800 red alleles in the population. The same thing done for the white allele. We have 20 homozygote recessive individuals, each having 2 copies of that allele, plus the number of heterozygote individuals that each have 1 copy of that white allele. This gives us 200 white alleles in the population. To convert these values into frequencies, we need to divide by the total number of alleles in the population. In this case, we have 500 individuals, each with 2 alleles. So we have 1,000 alleles total. When we divide our allele totals by the total number of alleles in the population, we get the frequency of each allele. So this tells us that 80% of our alleles in the population are red, while 20% are white. Notice that the sum of the 2 alleles will equal 1. It should always equal 1 because it's 100% of the alleles in the population. So wait a minute. Is this population evolving? Well, we're actually not quite sure yet. Now we need to use these allele frequencies and the Hardy-Weinberg equation to come up with predicted genotype frequencies. If I insert allele frequencies into the Hardy-Weinberg equation and then simplify it, we get the predicted genotype frequencies that we should see if the population is in equilibrium. We then compare those frequencies to our actual genotype frequencies we calculated at the beginning of this adventure. If they match, then the population is in equilibrium. However, if they do not match, then the population is out of equilibrium, gene frequencies are said to be changing, and the population is evolving. Here, they match, so the population is in equilibrium and is not evolving. Shifts out of equilibrium due to violation of those conditions can occur in lots of different ways. Here we have three primary modes of selection that can take place. We have directional selection, where one allele is favored over another, resulting in the favorable allele becoming more common. Then we have something called disruptive selection, where homozygous individuals for both alleles are preferred. This is really common when there's non-random mating occurring and individuals prefer to mate with others that look most like themselves. And then lastly, we have stabilizing selection, where the heterozygous individuals, those that have one copy of each allele, are favored and selected for. There are other factors that can influence allele frequency changes over time as well. Two specific types of selection can occur when available allele frequencies are changed due to sudden reduction in the number of individuals in the population. Let's look at bottleneck effects first. This type of selection occurs when a population is reduced down to just a few individuals. The reduction acts just like a filter, and only a few individuals are available for reproduction. Like in this cartoon, where each marble represents an individual with their alleles in the population are reduced when only a few individuals survive to the next generation. Diversity is lost in the process, and allele frequencies change. Large reductions in a population can occur due to habitat loss. Here we can see the bottleneck effect on a bird species called the greater prairie chicken. Originally, the distribution of these birds covered most of Illinois. However, urbanization and conversion of land for agricultural use dramatically reduced the population down to just a few small areas in the state. Scientists observing this bottleneck studied the allele frequencies and determined that the number of alleles was substantially reduced. One important additional note is that the reduction in diversity that occurred due to this bottleneck also resulted in a reduction in relative fitness. Fitness was measured by the number of eggs that hatched from this species, and it was significantly reduced when the number of alleles was reduced. This means that fitness of an organism was compromised by the reduction or change in allele frequencies and diversity. The founder effect is quite similar to the bottleneck. However, instead of reducing the population as a whole, a population becomes fragmented, and the new fragment has a different proportion of the diversity that was originally present. This is incredibly common when organisms colonize a new area like an island. The same thing can occur when we move wild populations into zoos for protection. The zoo population is just a fragment of the original population. This is why some zoos collaborate to interbreed their animals to maintain a healthy genetic diversity. The last two selective forces we will discuss are selections that depend on the frequency of the opposite allele. These selective forces are referred to as balancing selection, and we're going to discuss the heterozygote advantage and frequency-dependent selection. Have you ever heard of sickle cell anemia? It is a disease that results from a genetic mutation of a single nucleotide. Though it has a simple cause, it disrupts the morphology of the hemoglobin protein. This leads to a misshapening of the red blood cells and makes them less effective at transporting oxygen, as well as harder for them to travel through smaller capillaries. Sickle cell anemia is a devastating disease that can have systemic effects on the individuals that are afflicted. Fortunately, the disease only exhibits itself in an individual that is homozygous recessive. So, if an individual has only one copy of the damaged allele, they are a carrier for the genetic mutation, but do not exhibit the disease. Though it would be really hard to paint sickle cell in a positive manner, there is one innate benefit to the disease. A blood parasite called malaria that infects, reproduces in, and kills red blood cells is unable to effectively infect sickle red blood cells. This means the individual with sickle cell disease are much less likely to get malaria. Now, of course, they have sickle cell anemia, so that's bad enough. But something very fascinating occurs in someone who is a carrier for the sickle cell anemia, aka the heterozygote individuals. Having one functional allele and one damaged allele does not cause sickle cell anemia, but still provides some protection against infection from malaria. Those that do not have the mutation and are homozygote dominant are more likely to get malaria, and individuals who are homozygous recessive have sickle cell anemia. Thus, the heterozygote individuals have a distinct advantage over the other genotypes. So you may be wondering why all individuals are not heterozygous then. This is because heterozygous individuals still have the disadvantage of carrying one mutated gene that can be passed on to their offspring. And if they have children with somebody who also has a mutated version of their allele, then they can produce children that are homozygous recessive and have sickle cell anemia. The last mode of selection is a balancing selection called frequency-dependent selection. This works basically by one phenotype becoming more common in the population, and then there is more and more competition between those phenotypes. Thus, any individual that exhibits another phenotype would be favored. We can see this in the actions of this cute little fish. This Microevolution species we are looking at is actually a fish that feeds on the scales of other fish. They are born with their mouths facing either to the left or to the right, and they do not have the ability to move their mouth to the other side. This fixed mouth position means that the right-mouthed fish must always approach their prey from the left side, and the left-mouthed fish must do the opposite. As the population becomes increasingly homogeneous for one or the other, there's a strong selective pressure for the fish that can approach from the side opposite to all the others. Thus, allele frequencies change based on the frequency of the opposite allele in the population and oscillate back and forth. Well, that's it for this episode of Purple Bear Biology, y'all. I hope you found this exploration of microevolution useful. Drop a comment section below if you have any questions, and let me know what you think about the crazy cool world of selective pressures. As always, thank y'all for watching, and see y'all next time.